Hello, everyone. I will have to speed up just a little bit, but thankfully a lot of things have been already covered. The only things I will need to explain to you is why phosphorus MRS is a bit different from proton MRS, which my colleagues here have already talked about. So in order to do that, we need, even though some of you maybe don't want to, we need to look at the cell. This is a schematic representation of the cell, and it just so happens that phosphorus is involved in, let's call these two groups of processes, which are energy metabolism and membrane turnover. So let me just quickly go through them. The first cluster is actually oxidative phosphorylation in the mitochondria, which is the main source of energy in the cell, and as a result of this process, ATP is created, which hopefully many of you are familiar with, but this is the main energy carrier in the cell, and it is created with the help of NAD. And we will call this little cluster of molecules slow energy, because this process in cellular terms is indeed quite slow, but it is the main source of energy. And then the second one we have is phosphocreatine, so PCR, which serves as an alternative source of ATP, sort of source, in case mitochondria are not sufficient. So it serves as quick replenishment of ATP, which is why we will call it fast energy. And then finally, membrane metabolism. The two types of molecules we can look at with phosphorus MRS are phosphomonoesters and phosphodieesters, which are basically what precedes membrane phospholipids and what becomes of them when they break down. So this little cluster will be membranes, and I hope you don't get tired of these little guys in the corner, because they will be with us for a while. So just to sum up, again, ATP and NAD, energy, slow energy, phosphocreatine, fast energy, and then a membrane synthesis and membrane breakdown, membranes. So, why are we talking about this? We know that energy and membrane metabolism in the brain are involved in a multitude of different mental, neurological, psychiatric disorders or changes in this metabolism. And also, from the literature, we know that they can, markers of these types of metabolism can change with normal aging, can correlate to cognitive measures, other measures of brain structure and function like connectomes, etc. And they also can be quite different with sex. For example, ATP tends to go down with normal aging and also with neurodegenerative disorders, again, which seems quite obvious. While phosphocreatine, the hypothesis right now in the literature is that this might be a compensatory kind of mechanism. So while ATP goes down, phosphocreatine sometimes tends to creep up, again, because probably mitochondria are not sufficient, so phosphocreatine kind of needs to pick up its pace to compensate. And then the ratio between membrane synthesis and membrane breakdown also tends to go down with age, with different disorders, etc. Of course, that's not always the case, but this is just to illustrate that these types of metabolism are involved in a whole bunch of things. But how can we look at these metabolites? Mostly, what has been done is, again, like Joanne pretty much said, we either need to cut up humans or animals, but an alternative way is obviously MRS. Usually, it would have been done with the signal voxel acquisition, again, like already has been said by my colleagues. But nowadays, we can do multivoxel or whole brain acquisition, thus multiplying these types of spectra by the number of voxels and getting metabolite maps for each of the metabolites I had already mentioned. But why is this important? In the last couple of years, a couple of studies have come out. This is quite novel, to be honest, in the phosphorus space, especially compared to the proton. But still, there is some data that, for example, the ratio between ATP and phosphocreatine tends to be lower in the occipital lobe, and these ratios are also changing, and they have been found by several groups to be different, lower or higher in the temporal lobe. These are just examples, but these examples are pretty much it. This is pretty much what we know about the spatial distribution of these metabolites, again, because this is quite novel research. And also some of these studies that you see here have indicated that these spatial distributions may be dependent on age and sex. So, putting this all together, pretty much, about all of these metabolites, we know some things, but we know surprisingly little about their involvement in cognitive function. Because you can count on one hand the amount of studies that have been done kind of trying to correlate different cognitive measures to metabolites. And the ones that I've circled or squared here are the ones that have been that people have seen associations of with, for example, verbal learning. But what about all the rest of them? And also, what about the spatial distribution of these changes? Because those studies that have found these associations have been single voxel again. So to address all of these gaps, this study assessed regional differences in metabolism. So I will talk a bit more about in more detail. We also looked at the influence of age and sex, and then we studied the link between metabolism and cognition. So in terms of the methods, this study was done in healthy young adults, so 18 to 40 years old. Whole brain MRS was acquired for each of them. You might notice that the resolution of the of MRS is more than 2 centimeters. So phosphorus MRS is quite coarse, even compared to the proton MRS. So we need to account for that. And a convolutional neural network was used to quantify these metabolites and create metabolite maps. So in the end, we had 7 metabolites or metabolite ratios, which can give us kind of a different insight into brain metabolism. Simultaneously, all the participants completed the matrix consensus cognitive battery, out of several tests, we got 5 cognitive domain scores: visual learning, verbal learning, working memory, processing speed, and executive function, and also an average of all of these 5 global cognition, which were all assessed for correlations with metabolites. Just quickly, how MRS was processed. The metabolite maps were registered to the participants' T1, the T1s to standard space, which I'm sure you're all familiar with. Then the transformation of the first step was applied to, the second step was applied to the first step. The reason I'm actually drawing your attention to this is you might notice that in this process, the maps were resampled. So the maps you will see further are actually, like, they look like they are better resolution than they are actually, which also needs to be remembered. And finally, what I need to mention is that phosphorus, unlike proton, doesn't have an internal reference, so you cannot reference to water, because water doesn't have phosphorus. So what we kind of had to do, there are several ways, but we chose to reference all metabolites to the sum of all metabolites. So pretty much any results you will see here are like basically a percentage. So each metabolite is a percentage of the sum of all metabolites. And then in terms of the analysis, we did region of interest analysis to assess regional heterogeneity, so to compare regions with each other. And again, you might notice that the regions are quite coarse, because otherwise I felt like we wouldn't get reliable results because of the resolution. And even more of the reason why we needed to do voxel-wise analysis, which we implemented to look at age-related changes, sex differences, and also associations with cognition. But we will start with the regions of interest. So you can see here, these are maps of metabolite distribution, which again look nicer than the resolution before, but don't be fooled. So I guess what I would like you to take away from this slide in particular is that slow energy, so ATP and NAD, are quite similar with each other, as you can see. So they are lower in the temporal cortex, higher in the subcortical regions, like the basal ganglia, the thalamus, and the cerebellum. And then phosphocreatine, which is our fast energy guy, he is quite different, like pretty much the opposite to the slow energy markers. And then in terms of the membranes, these distributions are, first of all, it is interesting that the membrane synthesis and membrane breakdown are quite similar, so they're not like opposite or anything. But the distribution of PDE, so membrane breakdown, is a bit more homogeneous than the membrane synthesis. And now onto the most interesting part, which is the correlations, the voxel-wise associations with age and sex. So with age, we saw a correlation, like a negative correlation of NAD, which is not surprising at all. But then NAD hasn't been studied that much before, so it is interesting to see, especially considering the fact that we have a young sample, 18 to 40 years old. So even within that kind of limited age range, we already see a decrease in NAD, but not in ATP, for example, which has been known to decrease with age in a bit of an older sample. So this is, I think this is quite interesting, that probably maybe NAD is kind of more susceptible to age. And then we saw higher PME, so membrane, the marker of membrane synthesis in females, which I cannot talk too much about because this is very understudied, and a couple of the studies I saw actually showed the opposite effect. So this needs more thought put into it, but again, it is interesting. And now onto the correlations, so to say, with cognition. So for NAD and ATP to PCR, this is just a marker of energy demand. We saw positive correlations with verbal learning in particular. So this is kind of getting at mostly slow energy and then a bit of fast energy because there's the ratio to phosphocreatine. And it is interesting to know that these are correlations in the frontal, kind of, sorry, not in the frontal, in the parietal cortex mostly, and not, you know, in the temporal frontal, which would be more kind of obvious for verbal learning. And this is even more interesting, in my opinion, because phosphocreatine tended to correlate negatively. Again, so we're kind of sensing this trend of phosphocreatine being opposite, so fast energy being opposite to slow energy, again, probably maybe as compensation. So we found phosphocreatine to be negatively correlated with global cognition and verbal learning, again in the parietal cortex, which the parietal cortex may be implicated in kind of verbal functions in terms of like sensory motor combination. So maybe it was like, maybe it's due to the specific task that we implemented in verbal learning, which was kind of like trying to remember a list of words. But yeah, it is interesting that phosphocreatine is negatively correlated to it, which is novel in terms of the literature findings. It hasn't been reported before. So onto the conclusions. This study concludes that in young healthy adults, first of all, the distribution of phosphorus-containing metabolites is indeed heterogeneous, as you might have seen on the graphs. I forgot to mention that for all of the metabolites, the distributions were significantly different. They didn't just look different, they were significantly different for most regions with each other. And then there are sex and age-specific differences in brain metabolism, again, even in our limited, in terms of the age range sample. There are region-specific associations of energy metabolism with global cognition and verbal learning. And finally, obviously, this is a good note to end the symposium on, that it is important to prioritize whole brain analysis because if we did all the same things with just one voxel, we wouldn't have seen pretty much any of the results I showed you today, because this voxel would have probably been put in the frontal lobe, which was the case with the previous studies with phosphorus MRS, and then, yeah, we pretty much wouldn't have seen any of the things I have shown you. So some limitations of this study are that metabolic values, again, as I mentioned, are not absolute. The limited age range, and also the scans were registered to standard space using a coarse resolution structural scan, because it was done with the phosphorus coil, not a double-tuned coil. And some future directions are, we want to look at blood markers of mitochondrial function and metabolism, which would be very interesting because we got also samples in the same participant group. We got blood samples, and also from part of them we got CSF samples. So it would be like really interesting to ground kind of this imaging research in actual biological samples. And also we are planning to look at other measures of structural and functional connectivity and stuff. So, yeah, sorry if it was a bit rushed. I'm sending a special thanks to all of my supervisors, which one of them is in one of the neighboring rooms right now, and also to my dear colleagues and friends. Thank you.